I've talked a lot about the importance of hemoglobin in our red blood cells, so I thought I would dedicate an entire video to hemoglobin. One, because it's important, but also it explains a lot about how the hemoglobin, or the red blood cells, depending on what level you want to operate, know, and I have to use know in quotes. These aren't sentient beings, but how do they know when to pick up the oxygen and when to drop off the oxygen? So this right here, this is actually a picture of a hemoglobin protein right there. This is hemoglobin. Hemoglobin. It's made up of 4 amino acid chains. That's one of them. Those are the other 2. We're not going to go into the detail of that, but these look like little curly ribbons. If you imagine them, they're a bunch of molecules and amino acids, and then they're curled around like that. So this, on some level, describes its shape. And then in each of those groups, or in each of those chains, you have a heme group. You have a heme group here in green. Heme group. That's where you get the heme in hemoglobin from. You have 4 heme groups, and the globins are essentially describing the rest of it, the protein structures, the 4 peptide chains. Now this heme group, this is pretty interesting. It actually is a porphyrin structure, and if you watch the video on chlorophyll, you'd remember a porphyrin structure. But at the very center of it, in chlorophyll, we had a magnesium ion. But at the very center of hemoglobin, we have an iron. We have an iron ion. Iron ion. And this is where the oxygen binds. So on this hemoglobin, you have 4 major binding sites for oxygen. You have right there, maybe right there, a little bit behind, right there, and right there. Now, why is hemoglobin? Well, oxygen will bind very well here. But hemoglobin has several properties that, one, make it really good at binding oxygen, and then also really good at dumping oxygen when it needs to dump oxygen. So it exhibits something called cooperative binding. Cooperative binding. And this is just the principle that once it binds to one oxygen molecule, so let's say one oxygen molecule binds right there, it changes the shape in such a way that the other sites are more likely to bind oxygen. So it just makes it, one binding makes the other bindings more likely. Makes other bindings more likely. Now you say, okay, that's fine. That makes it a very good oxygen acceptor when it's traveling through the pulmonary capillaries, and oxygen is diffusing from the alveoli. That makes it really good at picking up the oxygen. But how does it know when to dump the oxygen? This is an interesting question. When to dump oxygen. It doesn't have eyes or some type of GPS system that says, oh, you know what? This guy is running right now, and so he's generating a lot of carbon dioxide right now in these capillaries, and he needs a lot of oxygen in these capillaries surrounding his quadriceps. I need to deliver oxygen. It doesn't know it's in the quadriceps. How does the hemoglobin know to let go of the oxygen there? And that's a byproduct of what we call allosteric inhibition, which is a very fancy word, but the concept's actually pretty straightforward. Allosteric inhibition. And when you talk about allosteric anything, it's often used in the context of enzymes. You're talking about the idea that things bind to other parts. Allo means other. So you're binding to other parts of the protein or the enzyme. And enzymes are just proteins. And it affects the ability of the protein or the enzyme to do what it normally does. So hemoglobin is allosterically inhibited by carbon dioxide, and by protons. So carbon dioxide can bond to other parts of the hemoglobin. I don't know the exact spots. And so can protons. So remember, acidity just means a high concentration of protons. So if you're in an acidic environment, protons can bond. Maybe I'll do the protons in this pink color. Protons, which are just hydrogen without electrons, right? Protons can bond to certain parts of our protein, and it makes it harder for them to hold on to the oxygen. So when you're in the presence of a lot of carbon dioxide or an acidic environment, this thing is going to let go of its oxygen. And it just happens to be that that's a really good time to let go of your oxygen. Let's go back to this guy running. He's running. There's a lot of activity in these cells right here in his quadriceps. they're releasing a lot of carbon dioxide into the capillaries, well, at that point they're going from arteries into veins, and they need a lot of oxygen, which is a great time for the hemoglobin to dump their oxygen. So it's really good that hemoglobin is allosterically inhibited by carbon dioxide. Carbon dioxide joins on certain parts of it. It starts letting go of its oxygen. That's exactly where in the body the oxygen is needed. Now you're saying, wait, what about this acidic environment? How does this come into play? Well, it turns out that most of the carbon dioxide is actually disassociated. It actually dissociates. It does go into the plasma, but it actually gets turned into carbonic acid. So I'll just write a little formula right here. So if you have a little bit of, if you have some CO2 and you mix it with the water, I mean, most of our blood, the plasma, it's water. So you take some carbon dioxide, you mix it with water, and you have it in the presence of an enzyme, and this enzyme exists in red blood cells. It's called carbonic anhydrase. Carbonic anhydrase. A reaction will occur that'll essentially you'll end up with carbonic acid. You'll end up with, we have H2CO3, and it's all balanced. We have 3 oxygens, 2 hydrogens, 1 carbon. And this it's called carbonic acid because it gives away hydrogen protons very easily. Acids dissociate into their conjugate base and hydrogen protons very easily. So carbonic acid dissociates very easily. It's an acid, although I'll write in some type of an equilibrium right there. If any of this notation really confuses you, or if you want more detail on it, watch some of the chemistry videos on acid dissociation and equilibrium reactions and all of that. But it essentially can give away one of these hydrogens. So you're left with, but just the proton, and it keeps the electron of that hydrogen. So you're left with a hydrogen proton plus, well, you gave away one of the hydrogens, so you just have 1 hydrogen. This is actually a bicarbonate ion. So this is bicarbonate right there, but it only gave away the proton, kept the electron, so you have a minus sign. So it's all of the charge adds up to neutral, and that's neutral over there. So if I'm in a capillary of the leg, let me see if I can draw this. So let's say I'm in the capillary of my leg. Let me do a neutral color. So this is a capillary of my leg. I've zoomed in just one part of the capillary. It's always branching off. And over here I have a bunch of muscle cells right here that are generating a lot of carbon dioxide. They're generating a lot of carbon dioxide, and they need oxygen. Well, what's going to happen? Well, I have my red blood cells flowing along. And it's actually interesting. Red blood cells, the capillary are 25%, their diameter is 25% larger than the smallest capillaries. So essentially, they get squeezed as they go through the small capillaries, which a lot of people believe helps them release their contents. So releases maybe some of the oxygen that they have in them. So you have a red blood cell that's coming in here. It's being squeezed through this capillary right here. It has a bunch of hemoglobin. And when I say a bunch, you might as well know right now, each red blood cell has 270 million hemoglobin proteins. 270 million. And if you total up the hemoglobin in the entire body, it's huge because we have, let me write this number down. We have 20 to 30 trillion red blood cells. We have 20 to 30 trillion red blood cells, and each of those 20 to 30 trillion red blood cells have 270 million hemoglobin proteins in them. So we have a lot of hemoglobin. So anyway, that was a little bit of a, so actually red blood cells make up roughly 25% of all of the cells in our body. We have about 100 trillion or a little bit more, give or take. I've never sat down and counted them. But anyway, we have 270 million hemoglobin particles or proteins in each red blood cell. Explains why the red blood cells had to shed their nucleuses to make space for all of those hemoglobins. They're carrying oxygen. So right here we're dealing with, this is an artery, right? It's coming from the heart. The red blood cell is going in that direction. And then it's going to shed its oxygen, and then it's going to become a vein. Now, what's going to happen is you have this carbon dioxide. You have a high concentration of carbon dioxide in the muscle cell. It eventually, just by a diffusion gradient, ends up, let me do that same color, ends up in the blood plasma. Ends up in the blood plasma, just like that. And some of it can make its way across the membrane into the actual into the actual red blood cell. In the red blood cell, you have this carbonic anhydrase, which makes the carbon dioxide dissociate into, or essentially become carbonic acid, which then can release protons. So let me do the protons out here. Well, those protons, we just learned, can allosterically inhibit the uptake of oxygen by hemoglobin. So those protons start bonding to different parts. And even the carbon dioxide that hasn't been reacted with, that can also allosterically inhibit the hemoglobin. So it also bonds to other parts. And that changes the shape of the hemoglobin protein just enough that it can't hold on to its oxygens that well, and it starts letting go. And just as we said we had cooperative binding, the more oxygens you have on, the better it is at accepting more. The opposite happens. When you start letting go of oxygen, it becomes harder to retain the other ones. So then all of the oxygens let go. So this is, at least in my mind, it's a brilliant mechanism, because the oxygen gets let go just where it needs to let go. It doesn't just say, oh, I've left an artery and I'm now in a vein. Maybe I've gone through some capillaries right here and I'm going to go back to a vein. Let me release my oxygen, because then it would just release the oxygen willy-nilly throughout the body. This system, by being allosterically inhibited by carbon dioxide and an acidic environment, it allows it to release it where it is most needed, where there's the most carbon dioxide, where respiration is occurring most rigorously, or vigorously. So it's a fascinating scheme. And just to get a better understanding of it, right here I have this little chart right here that shows the oxygen uptake by hemoglobin, or how saturated it can be. And you might see this in maybe your biology class. So it's a good thing to understand. So right here we have on the x-axis, or the horizontal axis, we have the partial pressure of oxygen. And if you watch the chemistry lectures on partial pressure, you know that partial pressure just means how frequently are you being bumped into by oxygen. Pressure is generated by gases or molecules bumping into you, or it doesn't have to be gas, but just molecules bumping into you. And then the partial pressure of oxygen is the amount of that's generated by oxygen molecules bumping into you. So you can imagine as you go to the right, there's just more and more oxygen around, so you're going to get more and more bumped into by oxygen. So this is just essentially saying how much oxygen is around as you go to the right axis. And then the vertical axis tells you how saturated are your hemoglobin molecules. This 100% would mean all of the heme groups on all of the hemoglobin molecules or proteins have bound to oxygen. 0 means that none have. So when you have an environment with very little oxygen, and this actually shows the cooperative binding. So let's say we're just dealing with, an environment with very little oxygen. So once a little bit of oxygen binds, then it makes it even more likely that more and more oxygen will bind. As soon as a little, that's why the slope is increasing. I don't want to go into algebra and calculus here, but as you see, we're kind of flattish, and then the slope increases. So as we bind to some oxygen, it makes it more likely that we'll bind to more. And at some point, it just, it's hard for oxygens to bump just right into the right hemoglobin molecules. But you can see that it kind of accelerates right around here. Now, if we have an environment with an acidic environment that has a lot of carbon dioxide, so that the hemoglobin is allosterically inhibited, it's not going to be as good at this. So in an acidic environment, this curve for any level of oxygen partial pressure, or any amount of oxygen, we're going to have less bound hemoglobin. So let me do that in a different color. So then the curve would look like this. The saturation, I want to do it darker. The saturation curve will look like this. So this is an environment right here. So this is an acidic environment. Maybe there's some carbon dioxide right here. So the hemoglobin is being allosterically inhibited. So it's more likely to dump the oxygen at this point. So I don't know how exciting you found that, but I find it brilliant, because it really is the simplest way for these things to dump their oxygen where needed. No GPS needed, no robots needed to say, oh, I'm now in the quadriceps and the guy is running, let me dump my oxygen. It just does it naturally, because it's a more acidic environment with more carbon dioxide. It gets inhibited, and then the oxygen gets dumped and ready to use for respiration.